No, I think we might get started actually. We've got a great turnout today. It's really wonderful to have you all joining us. And I am really delighted to introduce Christine Sprunger, who is an assistant professor at the Michigan State University, based at the Kellogg Biological Station. I was lucky enough to hear Christine present when she visited UW-Madison. I think that might be 2 years ago by now.
Yeah.
And I just really enjoyed learning about her work on different tillage systems used by organic farmers and learning some really fascinating details about what we can learn from the lives of nematodes in the soil and how they can help us understand the health of the soil. So I'm sure you're going to really enjoy this presentation, and I'm going to hand it over to Christine.
Great. Thank you so much. It's a pleasure to be able to share my work with all of you. If you have questions during the talk, please just put it in the chat and Rue will signal to me that I need to halt and I'll try to answer your questions. And I'm looking forward to a lot of discussion at the end. So the title of my talk is Soil Health, Nutrient Cycling, and Soil Biology in Reduced Tillage Systems. So I'm sure all of you have heard about soil health. It's definitely a buzzword that gets thrown around quite a bit. And in recent years, it's really gained a lot of attention, not just from farmers and researchers, but also policymakers. There's always new kind of funding opportunities around soil health. And I think that's really because there's a strong understanding that soil health is so interconnected to agronomic productivity as well as important ecosystem services. But just to get everyone on the same page, the most common definition for soil health is that it is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. But what does that actually mean from a practical standpoint? When we think about the way that we test soils, in the past, it's been that we largely focus on our chemical indicators. And what soil health does is it actually allows us to think about soils through a more holistic lens. And that means we're also considering the biological and physical components. And what's even more important around the aspects of soil health is through intensive research, we've come to realize that soil health is critical for enhancing biological diversity. We see critical linkages with food production. There's also important connections to water benefits as well as carbon storage. There's also this recognition that soil biology is extremely important for soil health. And I really want to bring this point out because, again, a lot of the indicators that we have used in the past for quantifying soils and even soil health have largely been focused on chemical and physical indicators. And we know that the soil ecosystem is super complex. It serves as a habitat for all sorts of soil organisms. And we're just now understanding how we can kind of integrate different soil organisms and use those as bioindicators of soil health. And I'll definitely talk more about this in the latter portion of my talk, but I really just think it's important to center soil biology within the context of soil health. It's also important to think about soil health from a local and regional kind of perspective. I've been working with farmers on all aspects of soils and soil health for the past 10, 12 years. And one thing that has really struck me when talking to farmers is that farmers want more information from what current soil testing standards are offering. So, when you send your soils to a commercial testing lab, you oftentimes just get total organic matter, and then you get a lot of different chemical processes. And what can be frustrating sometimes is that those indicators take a long time to change. And so, maybe you're testing every 3 years, you might not see differences over that 3-year period, which is frustrating if you're working towards incorporating different management practices, incorporating regenerative agriculture. You want to make sure that your hard work and efforts in incorporating these new managements are actually leading to better soil health. So because of that, we've seen a lot of enthusiasm for soil tests that are more sensitive to recent changes in management. And so that is like one major motivator for why we need to increase soil health assessments, especially across the Midwest. There's also this pressure to improve soil health from a water quality standpoint. So we're all in the Great Lakes region. We understand that there's a need to maintain and improve our water quality. And what we're trying to do is also understand the linkage between soil health and nutrient retention. So, where the field of soil health needs to expand and improve, in my in my personal opinion, is I think we need to do a stronger job of understanding how soil health responds to management, both using university experimental trials, but also working directly on farm and partnering with farmers. And so you'll see kind of two examples of this in this talk. We're, as scientists, working to rigorously vet soil health indicators. So what does this mean? We want to make sure that we're getting accurate information to our users. And so that means we need to make sure that these indicators are accurately predicting and quantifying soil health. We also want to do a better job of, once we quantify these soil health measures, we want to make sure that we can develop sound management recommendations for farmers. And that is definitely still a work in progress. We're also really working to integrate soil ecology and soil biology into the soil health framework. Like I mentioned briefly, we rarely see direct measures of soil organisms in our soil health frameworks. And so my lab has been kind of instrumental in trying to integrate nematodes into the soil health framework. Again, I'll talk a lot about that over the course of this talk. And then one last thing that we're working on in my lab is really thinking about soil health assessments for climate adaptation and mitigation. And I'll talk a little bit about that. But if you have questions around that, please, I would be happy to discuss that at the end. So when we think about soil health, it's important to think about, well, what management practices may actually lead to improvements in overall soil health. And here are just some guiding principles that we can use. So first, it's really important to always increase organic matter inputs. We see this as a really strong driver of soil health improvements. And you can do this in multiple different ways. You can move from using a tilled system to moving to no-till. We know that that increases organic matter over time. You can work to incorporate cover crops into your rotation, or even just diversifying your rotation by not just growing corn and soybeans, but also including small grains. It's also really important to think about diversifying our soil biota with plant diversity. So again, that gets to crop rotations and thinking about incorporating perennials perhaps into your rotation. That ties directly into also wanting year-round ground cover. So there is a correlation with the more bare soil that you have within your rotation, the likelihood that you're going to have decreased soil health. So again, if you can have cover crops during the off season, if you can incorporate perennials into rotation, those are all going to be really important for enhancing soil health. And then lastly, it's really important to think about living roots throughout the year. And again, that ties into everything I've just mentioned in terms of wanting to have roots of cover crops and roots of perennial systems, especially throughout the year, because those are going to be feeding your soil and really boosting your soil organic matter. But even though we have these kind of soil health promoting practices and guidelines, until recently, we actually didn't know if soil health promoting practices actually increase soil health. So we used a long-term trial that was established at the Kellogg Biological Station here at Michigan State University in 2009. And we wanted to see how different management practices actually led to soil health improvements. So this long-term trial goes from a low diversity gradient to a high diversity gradient. So there's a continuous corn managed through no-till. There is a corn, soy, wheat, or just corn, soy rotation plus a cover crop. And then there are monoculture perennials, switchgrass, miscanthus, poplar. These are candidate biofuel cropping systems, but also serve as crops that reflect what other monoculture perennials might be doing in the landscape, things like alfalfa, for example. And then we have our polyculture systems, things like native grasses, early successional and restored prairie systems, which really give us a great understanding of how like grass mixes especially might be helping with soil health. And just one note, this poplar system, it is a monoculture tree crop, but it has a vigorous understory. So we tend to count this more as a polyculture system as well. So the next thing we have to ask is how do we actually quantify soil health in these systems? And again, there are numerous ways and numerous indicators that you could use to quantify soil health. But here we really wanted to focus on measures that really reflected biological activity. So we focused on things like microbial respiration. This is really getting at CO2 production from microbial activity, and that is a really great indicator of the amount of carbon that's in your given system. Then we looked at active carbon, or also known as permanganate oxidizable carbon, and this is a indicator that we've now learned is reflecting more of a process pool of carbon. And all that really means is it's doing a really good job of showing us kind of early indicators of carbon, long-term carbon storage. And then we have soil protein, which is showing your organically bound pool of nitrogen. And it's really strongly tied to soil organic matter. So what did we find in this long-term trial? And we sampled this 9 years post-establishment. So the way to read these graphs is in gray, we have an annual system. In orange, we have a monoculture system. And then in tan, we have our polyculture perennial systems. And so what we can see, if we just focus on graph A for a moment, we see that our polyculture and monoculture perennials have about 1.5 to 2 times more microbial respiration relative to the annual system. So this is showing us that perenniality and diversification are critically important for boosting this important soil carbon pool. When we look at graph B, we still see differences across these different systems, but the magnitude between especially annuals and perennials is reduced. So this is, again, why it's important to have several different indicators that reflect different parts of your soil organic matter pool, because this, again, this indicator is showing, more processed pool of carbon. And this just means that it takes longer for this specific pool to change. So if we came back, after 15 years, we might start to see even larger differences between the annuals and perennials. When we look at the soil protein, again, this is the organically bound pool of nitrogen. We see a very similar result to microbial respiration, where we see pretty strong differences between our polyculture perennials relative to our annual systems. And again, this is another, just example of having our diversified crop rotations, and especially incorporation of perennials as boosting overall soil health. So just a quick summary here. We can conclude that systems with greater perenniality and crop diversity increase soil biological health. Perennial polycultures, aside from the restored prairie, also increase that active carbon pool, or POC, relative to annuals, which has important implications for long-term carbon storage. We also saw that rotated annuals with a cover crop actually increased active carbon relative to continuous corn. And soil biological health indicators have the potential to serve as indicators of both nutrient availability, especially that organically bound pool of nitrogen, and soil carbon stabilization, which again is critically important for long-term soil health and long-term carbon accumulation. So the example I just gave was really just focused on a long-term experimental trial. And we know that, when we focus on long-term trials that are, perfectly manicured, they're a controlled setting, it doesn't always reflect what we see in the real world. And that's why it's so incredibly important for us to really ask, what drives soil health on actual farmer fields. Not only is this important because we want to be able to really get this information out to farmers, but it's also important to scale up these results. Looking at one soil type in one location is just not the same in terms of fully understanding the potential for soil health across our agricultural landscapes than really scaling up and looking at farms across the Midwest. So to do that, we actually teamed up with a group of social scientists that were sending out a survey to every organically certified corn grower in Ohio, Michigan, Indiana, and Pennsylvania. And we decided that we would offer free soil health tests to any farmer that completed the survey. So we just sent this postcard along with that survey, and then farmers, if they were interested, they would send back the postcard, and then we would send back instructions for how to sample their farms. And so what we asked farmers to do who participated in this study was to select a soil sampling area that was 5 acres or less, and then take cores or slices to a depth of 20 centimeters. And then we asked them to send a composite soil sample, about 3 cups of soil, with an additional management survey, because we really wanted to know exactly what farmers were doing on these specific fields, so we could then relate it to different soil health metrics. So when we received the soils, we sieved the soils to 8 millimeters and then ground them to 2 millimeters, which is a standard protocol for soil health assessments. And then soils were analyzed for chemical, physical, and biological properties. And then once we were done, we sent a comprehensive soil health test to every single farmer that sent in a soil sample. So what did we find in terms of management? So management surveys, this is just an example of how we ask for management survey data, we asked for about 4 years of management history. And this is just an example. A farmer had 3 years of hay and then corn and then 3 years of hay. We also knew that it would be important to quantify tillage. And so we wanted to understand both the type of tillage as well as the frequency of tillage. Again, these were all organic corn growers. So in terms of the number of samples and just state and county representation, we had 4 states, 73 counties represented, and ultimately ended up with 195 soil samples. So a quick summary of the management practices represented in the study. We found that 87% of the farmers were adding some type of manure. We found that about 47% of farmers were incorporating cover crops. And we found that 76% of farmers were incorporating a perennial, which to us was a really surprising finding. And then for tillage, we found that 92% of farmers were incorporating tillage. And you see that 8% there, you're like, well, how are they growing organically certified corn without any tillage? This was likely those farms that had 4 years of perennials in their rotation. The other thing that we're able to do with this is really ask, what is a good soil health test? So the next figure that I'm going to show is a bit overwhelming, but it is a really important kind of practical point that I want to make with this. Until this specific study, we had worked with farmers on soil health assessments. In Michigan, we worked with about 15 different farmers. And when we showed farmers their soil health test reports, they would always ask, well, is this a good soil health value or a bad soil health value? And the truth is we couldn't actually answer that because these soil health metrics were so new that we didn't have a representative sample to be able to say, yeah, for your soil type, this is a really good value. And so with this study, getting 195 samples from across the Midwest, all sorts of soil types represented, we are now able to say, yes, this is a good soil health test relative to farms with a similar soil type. And the way that we were able to do that was we were able to come up with distributions for every soil health indicator that we measured. And again, this is a lot to look at. So let's just look at POC. So this is the same indicator that I just talked about in the first part of the talk. And what we can see here is that we looked at this value based on 3 different kind of soil textures: coarse, medium, or fine. And these dotted lines represent the average. And so we were able to now say, if you have a value of 600 for POC, you're going to be above average relative to your soil type. So again, it's just a really complicated way to say that we now have a way to demonstrate where farmers are at in terms of their soil health reports, which we think is really important for being able to then implement various management practices based on soil health scores. So the next thing we want to ask is, what's the relationship between these different types of management practices? We were looking at tillage, we were looking at crop rotational diversity, we were looking at perenniality, and we thought it'd be important to really understand the relationship with these different types of management practices. So what we noticed right away was there is this really interesting correlation between crop diversification, tillage, and perenniality. So in graph A, we're looking at the relationship between tillage frequency and crop diversity. And crop diversity was just calculated looking at the total number of crops in a given rotation. And what we see here is that tillage frequency actually increases with crop diversity. So this tells us that farmers who have more crops that they're trying to work into their rotation, they're having to till more. But of course, when you have perennials in your rotation for a longer period of time, then we see a negative correlation where you see a decrease in overall tillage frequency the longer that you have perennials in your rotation. So now that we understand kind of the relationship between management, we wanted to directly ask, how do management practices influence soil health in organic corn production? So just looking at the 3 soil health indicators, again, these are the same ones that we talked about in the first part of the talk, we actually see that there's a slight negative relationship between the soil health indicators and crop diversity. So this means that soil health indicators are actually decreasing with an increase in crop diversity. This was a shock. If you remember in the first part of like the first or second slide, we talked about how important crop rotational diversity is for improving soil health. So when we saw these values, we knew we had to dig in to the data a little bit more to understand what is actually happening. When we look at the relationship between tillage and soil health, we begin to kind of understand why we're seeing decreases with crop rotational diversity. We see that there's a significant decrease with an increase in tillage frequency. So we kind of think that tillage frequency is really the driver of reducing overall soil health. We also wanted to look at the soil health indicators in terms of perenniality. So the x-axis here is perennials present in crop rotation by year. So for example, if a farm didn't have any perennials in the rotation, that's 0. If there were 4, that number 4 represents 4 years of continuous perennials in their rotation. And you can see that the longer you have a perennial in your rotation, the greater your soil health value. So what does this all mean? This is one of the first studies to explore that management impacts on soil health indicators using a large mail-in survey approach. We found that crop rotational diversity has a complex relationship with soil health indicators, most likely due to increased tillage frequency. When we see an increase in crop rotational diversity, we also see an increase in tillage, which leads to a decrease in soil health. When we see an increase in the length of perennials that are present in your rotation, you see a reduction in tillage and an increase in soil health. So this research really highlights the importance of conducting on-farm research. I think before this study, we didn't recognize the frequency that certified organic corn growers were using tillage. And I think it really demonstrates key trade-offs that happen, especially on farm. And it's really just important to use these large surveys to assess soil health. Okay, yeah, I see a question.
Yeah, Christine, one of our attendees had a question about the R values in your graphs showing correlations, saying that they seemed low, and could you talk about the strength of the relationships that you observed?
Yeah, that's a great question. So for soil health values, we generally see correlations anywhere between 2 and upwards of 0.6 or 0.8. So that negative 0.32 was a significantly strong correlation. And so that was a pretty strong negative relationship between mineralizable carbon and tillage frequency. The others, 0.02 and 0.06, are extremely, extremely weak, and these were not significant. So that's a really good point to consider.
And I just raised my hand again because I know that for those who aren't used to thinking about statistics, R value is just like, what do you mean R value? And Christine, you can certainly say more about this, but I was just going to say I very much understand the perspective of looking at a cloud of dots and thinking, how did you get a line out of that? But yeah, these numbers do give us a pretty good indication of what the overall trend is in what looks like a cloud of data. But did you want to say more about that, Christine?
Sure. Yeah. So, one of the main goals that we're trying to do is understanding the relationship between two response variables. And one way to do that is looking at correlations. And correlations are really just looking at how closely related two variables are. And we can do that by calculating the slope of the line, and that's what that line represents. And that R value tells us the strength of that relationship. And so when we see a downward trend, that shows a negative relationship. And when we see an upwards trend, that demonstrates a positive relationship. So here we have two graphs that are trending downward, showing a negative relationship. But that one in the middle is a significant negative relationship, which means we can confidently say that tillage frequency is negatively impacting mineralizable carbon. Any other questions before I move on?
Yeah, would you mind going back to that slide?
Sure.
Thanks. I think some of it's just some confusion I have always had about the mineralizable C versus the epoxy.
Yeah.
And I'm wondering, so that's just step one, and why the epoxy seems more closely related to protein. And then also, if you can, I'm not going to hold you to it, but like if you have a theory about why there's a strong correlation in one case and not in the others.
Yeah, that's a fantastic question, and the answers relate to one another. So, yes, the difference between mineralizable carbon and epoxy is pretty confusing, but I'm going to tell you how we measure them. So hopefully that helps you understand a little bit more about why these indicators are both telling us about carbon, just different parts of carbon. So if we think of the total soil carbon pool, so say you get your soil organic matter value back from your commercial testing lab. That is looking at the total pool of carbon. But like I mentioned, there are so many different things happening within the soil, right? There are microbes that are respiring and emitting CO2. There are other carbon molecules that are closely tied to the minerals in the soil. All of these kind of carbon pools are acting very differently. So one way to kind of guess how they're acting is based on how long they're actually going to persist in the soil. So the mineralizable carbon pool, we know reflects carbon that's really persisting in the soil for about a day. And that's because it is so easily accessible by microbes, that microbes can just eat it up, use it as energy, and respire it. There's other pools of carbon that are more physically protected from microbial activity that are going to stay in the soil longer. And that is where that epoxy value is coming from. And so that when you see epoxy, you can say, oh, that carbon is just sticking around a little bit longer. It's not as accessible to microbial communities. And it's, still very highly reflecting my overall total organic matter value. So what this means is that mineralizable carbon is more sensitive to recent changes in management. And so that's why I think we're seeing such a stark difference with these lines, is that mineralizable carbon, because it's more sensitive and more available to microbial activity, that also means that it's going to be more sensitive to disturbance and other management practices. Whereas epoxy, it's not going to be as responsive, especially right away. Does that help at all? Great, thank you for the question. All right, and happy to talk more about all of these different indicators at the end too, because there are so many, and that is part of the trick to understand how do we quantify them, how do we relate them together to actually tell the story about soil health. So this last little story that I want to tell is really asking, how does biology fit into the soil health framework? And the way that my lab does this is through thinking about nematology. And so oftentimes you probably, whenever you think about nematodes, you likely think of parasitic nematodes, soybean cyst nematodes. That's certainly a part of the nematode community, but there are actually beneficial nematodes that are really critically important for soil health. So I'll go into that really clearly in the next slide. But just taking a little bit of a step back, soil health really needs to address biodiversity. In the first couple of slides, I mentioned that biological processes are critical to soil health. And until recently, we haven't really done a good job of quantifying actual biodiversity. We have all these different indicators that are reflecting microbial activity and nutrient cycling, but we haven't really directly measured organisms in the soil. And we have actually been directly asked by a lot of farmers to measure organisms. So when I worked on farms in Michigan as a grad student back in 2013, farmers were looking at all these values and saying, yeah, I recognize that these are reflecting biological activity, but why aren't you actually directly measuring organisms? So that really got me thinking, and I thought, maybe nematodes could serve as an informative biological health indicator. And so why nematodes? So this is a broad kind of overview of the entire soil food web. But if you see right in the middle, you see the nematode communities. So nematodes are specialists. They're microscopic roundworms, and they're doing a lot to kind of influence the overall soil food web. So you have some nematodes that are the plant parasitic nematodes. There are soybean cyst nematodes. There are nematodes that feed directly on plant roots. Then you have nematodes that only feed on fungi. We call them fungivores. Then you have nematodes that are bacterial feeders. So they feed on bacteria, and we call them bacteriivores. And then you have actual predators, and those are nematodes that feed on each other. And based on the ratio of these different types of nematodes, we can actually tell a lot about overall soil health. We can, if we see more bacteriivores in a system, for example, that can be an indication of more disturbance. If we see more fungivores or predators in a system, that can be an overall indication of more nutrient cycling and overall stronger soil health, for example. So the way that we sample and for these nematodes is we take a soil sample and then we put them in these Bierman funnels. That's on that, if you look at the most left graph, we put them in funnels. We water those funnels every day, and nematodes are so attracted to water that water pushes the nematodes down towards the bottom of the funnel. And then after a 3-day period, we release the funnels and then we collect the nematodes. And then we're able to count them and then use a microscope to identify them. So the study that I want to talk to you about today is really asking how do nematode communities respond to agricultural intensification over extended periods of time. So I'm going to give an example from another long-term trial that's based here at Michigan State University at the Kellogg Biological Station. This was a trial that was actually started in 1988. And in 1991, a really famous nematologist, Diana Wall, who's at Colorado State University, sampled and quantified nematode communities. And then when we're looking back in the data archives in this published paper, we realized that no one had looked at nematodes since 1991. So my grad student, Tavisha Martin, decided for part of her PhD that she'd go back and carefully look at the exact measurements and protocols that Diana Wall used. And then we would go back and sample in 2021 and make a comparison. So what do we see? So these are stacked bar graphs, and these are just showing the overall feeding groups and their abundance in a given soil sample. So in green, we have the plant parasitic. In dark brown, we have the omnivores. So actually some nematodes can change their feeding preferences based on the environment. So those are, that's what an omnivore is. And then a predator is the tan bar, fungivore is the dark gray, and then bacteriivore is blue. And then when we look across this gradient of the LTER, from left to right, we have a conventional system, we have a no-till system, we have a reduced input system, we have a biologically based system that only receives a cover crop. We have a poplar system, a switchgrass system, an early successional, and a mown grassland. So it really goes from like conventional annual systems to perennial polyculture systems. And when we look at the 1991 data, we actually don't really see much of a difference. We see that all of the systems are dominated by bacteriivores and plant parasitic nematodes. But when we look at 2021, we start to see that these bars have kind of shifted in terms of proportionality. So the dark gray, the fungivores, there are much more in 2021 versus 1991, which tells us that the community has shifted over these 30 years. And one thing that's really interesting is when you start to see greater fungivores in your systems, that generally leads to or is associated with greater overall health in your system. Another way to look at this is graphing out a principal component analysis. So in the most simplest terms, the dots that are close together are systems that have nematodes that are very closely related to one another. And then when you have a dot that's further away, those nematode communities are very distinct from the rest of the systems. So in 1991, you had the mown grassland, which is a system it's never been tilled. It's this very conservation reserve area here at KBS relative to all of the row crop and perennial systems that are closely tied together. That means that the nematode communities were super similar, but very different from what we found in the mown grassland. But in 2021, we see something completely different. So we actually see that most of the systems, the treatments here, are moving towards the mown grassland. And really what we see is the conventional system is off on its own. So what this is showing us is that when you have consistent long-term management where you're incorporating no-till, where you're using cover crops, where you're incorporating perennial systems, you start to have a healthier and more balanced soil food web because the nematode community is really converging with that conservation reserve treatment. So just in summary, we feel that nematodes are strong candidate soil health indicators because they respond to contrasting management. Nematode quantification is really critical for assessing trophic guilds. So really all that means is that they're really important for getting a sense of overall soil biodiversity in a system. And we even see decadal changes in nematode community structure, which demonstrates that regenerative agriculture practices can lead to healthier soil food webs. So just some overall conclusions from this talk. Systems with greater diversity and perenniality enhance soil biological health in long-term trials in Michigan. On-farm soil health assessments will be critical moving forward as we work to incorporate soil health data into management recommendations. And nematodes should be included in future soil health assessments because they reflect key biological processes in the soil and clearly respond to contrasting agricultural management. So at this point, I'd just like to thank you all for listening and acknowledging everyone in my lab. They're the folks that are doing a lot of this hard work. I have my email and website listed here. Please reach out to me if you have any more questions, and also thanking my funding sources. But excited to chat more with you all right now. Thank you.
Thank you so much, Christine. That was really wonderful and fascinating. So many details there that we could dig into. So thank you for sharing. I'm gonna ask if anyone has questions. Please feel free to come off mute, and or I see your hand, Lincoln. So yeah, just in general, raise your hand virtually or on the screen. And if I don't notice, please feel free to come off mute. But Lincoln, why don't you go ahead?
Hi, that was so fun. Thank you. Could you go back a couple slides in the nematodes? Because it looks like it was the relative abundance, and I'm just curious, like, did overall abundance increase, or am I not thinking about nematodes the right way? Like, I tend to think that more of a thing is better, but maybe I'm like on the wrong track with that. So I'm just
No, that's a great question. So the reason why we wanted to look at relative abundance was because that was the calculation that 1991 used, but absolute abundance was very similar in terms of these overall trends. And so generally what we see is early on, we have greater overall bacteriivores, and then when we move into 2021, there seems to be a shift in the community. We have more fungivores, less plant parasitic nematodes, which is always a really good thing to see.
Also,
I have a follow-up question that's kind of related. It looks like the no-till segment is the only one that didn't increase its fungivores. So like,
no, so that is a really great question, and I want to go to this graph in particular because the no-till, while it's trending towards the mown grassland, just like the other systems, it's still kind of off on its own. And one of the things that we're kind of investigating right now is that the no-till system receives the largest amount of pesticides of all of these treatments. And so we're wondering, do the pesticides and insecticides that are applied, does that directly impact the overall health of the nematode community, specifically within the no-till plot? And I think that could be why we see less fungivores. So again, trade-offs that happen within these regenerative ag systems. And we're actually my grad student is looking at a specific experiment where she's applying different types of insecticides and nematicides and looking to see what happens to the overall nematode community. So really great observation.
I'd like to ask a question that follows that up a little, Christine, and also relates to the research that you've done to assess soil health on farm. So most of the farmers who have joined us here today are diversified vegetable farmers and farming organically. And I think that, just what you said about the limitations of some of these long-term trials, in some cases, they give us really amazing information on trends, but they can't possibly reflect everything that is going on out there. So in terms of assessing the relative proportions of these different members of the nematode communities from on-farm sites, do you feel that similarly to your soil health measures of, this is a good value, this is a less good value, how close are we to being able to take a sample from a farm site, look at the proportions of these nematodes, and say something meaningful about soil health?
Yeah, great question. And we actually have a study where we're doing that right now. So we're working with several farms in Michigan, and then we've just been recently connected to farms in Vermont and New York, and some in Idaho, and they've been sending us soil samples. And so what we don't have is a database that now, let me just back up for a second, because this is really important. So for our overall soil health assessments like respiration, active carbon, and soil protein, I showed the study where we had 195 samples, but really over the course of my, 12 plus year career, we've collected over 2,000 samples. And so we're able to have this really strong database of soil health that can serve as a baseline for large parts of the Midwest. We're not quite there yet with nematodes. We probably have about 50 samples that are from actual farmer fields. And so what we're trying to do is learn from these initial on-farm samples and really look at how the management of different farms is influencing soil health values. So what I can tell you is that the farms from New York, they're all vegetable farmers, and they're all certified organic. And we're actually seeing more predators than what we see in the corn based systems here in Michigan. So again, those are just kind of observational for now, but that shows you that organic, smaller farms, vegetable farms seem to have better overall soil health from a nematode perspective.
That's exciting. Thank you.
And how, I have two follow-up questions there, because I think Rue was kind of getting to the point for those of us who are like, what am I, how can I? Three, actually. So the first is, I guess, how responsive, this seems sort of exciting because I assume they have a pretty short life cycle, and it's something that you could track system A versus B. And so the second thing is like, is this a thing that's available? And the third is we've been kind of pushed in like a PLFA direction for diversity. And I've just said yes, because I'm not sure what else really to hold up against it. And so I just wonder if you have thoughts about PLFA versus this, or yeah.
Yeah, all great questions. So the first, yes, nematodes turn over really quickly in the soil. And the limitation with nematodes is that you're literally looking at a microscope and identifying nematode after nematode. So it's super labor intensive. And my grad student who's been identifying nematodes now for 4 or 5 years, she can do one sample in 10 minutes, But when she was just starting out, it took her an hour to do one sample. So it's laborious regardless of if you are an expert or not, but if you're just starting out, it's even more time consuming. And the issue is that there are probably 4 or 5 university labs across the country that actually know how to identify beneficial nematodes. So your next question, is it available? We want it to be available because we want to build a database that demonstrates how nematodes are responding to management on actual farmer fields. So we are about, I would say, 2-3 months away from opening up a soil health testing lab where farmers can send in their soils for a fee, and then we'll send them all of the soil health metrics that I showed in my talk. Your question about PLFAs, yes, that has been, I think, likely the most common kind of way to get at organisms within the soil health framework. PLFAs are great for being able to characterize the community, but from a, I don't know, I guess like mechanistic scientific kind of approach, we know that the better way to get at the microbial community is through expensive shotgun metagenomic protocols. And so the issue with all protocols that get at directly measuring organisms is they either take a lot of time or they're really expensive. And so there are kind of trade-offs to all of them. What we really like about nematodes over PLFAs is we've been able to really relate the nematode community more closely to these other soil health indicators. I didn't really go into that today, but they're strongly related to things like epoxy, soil respiration, whereas it's harder to get that connection with the PLFA data.
Lincoln, I'm going to put you on the spot and ask you, unless someone else has a question right now, I'm going to ask you to just briefly mention that you're doing some research with farmers in your region. Can you fill people in a little bit?
Sure. Is now it's fine?
Yeah,
I guess.
take a minute.
Yeah, well, we have been doing, I mean, I have a small diversified veggie farm, and we were doing a lot of experimentation with growing cash crops in a perennial Dutch white living mulch, Dutch white clover living mulch. That was exciting, but you know, it's just one farm, and we really wanted to see what people's experience was with this, and also just problem solve together. I mean, I'll be lucky if I even get 20 more seasons, and that's not a lot of experiments to run. So, you know, wanting to have 20 or 30 or 40 or 500 experiments running every year so we could learn faster. So we're funding trials now. So we started an organization called Momentum Ag, and we're funding trials on 20 farms this past season, mostly in the Northeast. There was a couple out in the Midwest, but mostly Massachusetts, Vermont, New York, Connecticut. And if anybody's interested in participating, you can go to momentumag.org and fill out a little very brief thing there for more information about enrollment. But yeah, we're just trying to create a community of farmers that can problem solve this stuff together. And, you know, this kind of work, I think, Christine, is so interesting because, I mean, this nematode thing is really exciting because it's so hard to get at these questions. And often it feels like the data we gather is just so we can do grant reporting and not really learn or like scratch our heads. And this just, you know, the idea that this would be closely tied to other soil health indicators. And I take your point that it's very labor intensive, but at least you're starting from a point where you feel confident that if you see these communities increase overall and you see their relative composition move in the right direction, that means good things are happening in the soil. And I think, yeah, it's just, I think part of the goal of our organization, Rue and I have talked a lot about this, is like, what's the information that farmers want to know? And let's make sure that we get that first. And a lot of that's really basic management questions that often are like equipment questions or seeding rate questions, or it sounds it's like not complicated. It doesn't yield itself to graphing, but it's like, these are kind of like step one, like, can we make it work? And then we also want to be kind of, what was Ronald Reagan's thing? Like trust and verify, right? Wasn't that? So, I trust that a system like the one we're working on that involves a perennial clover living mulch is a good thing. It's really important to verify. And like Christine, when you're showing that slide of sorry, I'm blanking on the inverse relationship there between diversity, between crop rotation diversity. Yeah, there you go. Sorry, it was right where you were on the other one. Oh, right. And just being like, oh, what that means is more tillage passes actually. This is why we verify also. Like there may be other things, it's just important not to just march ahead with it. But anyway, yeah. So trying to get that mix of like some targeted data collection that we really feel is valuable beyond just grant reporting, but really means something real. But then at the same time, really focusing on farmer-led research.
Thank you so much for
A lot
sharing.
to say.
That is super fascinating. And I love that because yes, we need so much more on-farm research and we need it to be applicable to farmers. And this specific study was getting at certified organic corn growers, but a lot of them actually were Amish growers and they were doing a lot of vegetables, etc. And I think that is why we saw, when you start to see like 7.5 crops within a 4-year rotation, like they were pushing and getting, vegetables, cover crops into their rotations. But unfortunately, they just had to till to get those different vegetables and different crops into their rotation. So I hope that this can be an informative way to demonstrate trade-offs that happen in our agricultural systems. But can I ask all of you a question? I would love to know, what, soil health metrics or agronomic metrics, what's most useful to all of you? We've had conversations with farmers, we've interviewed farmers about soil health tests, but it's always so helpful to get feedback and input from farmers. Like, are there a specific metric to you that seems important that maybe we're missing or not really thinking about? I'd love to hear your thoughts.
I guess I'll go. Hello, can you guys hear me?
Yes.
Yeah, okay. Yeah, I guess I'm really just interested in the biology of the soil, what's actually in there, just starting to identify what sort of creatures I do have. I feel like we've focused so much on compost the last couple of years, and I've seen such a dramatic improvement on the crops that are growing that I'm not as much interested in the chemical makeup of the soil, because I just feel like everything is there that needs to be there probably for the most part. And so then I'm just really, and I obviously that's a real big broad stroke, but I'm really just interested in the life that's in the soil and just starting to categorize and capture what we actually have in there. And then I've been kind of a little bit skeptical of like bringing in nematodes and other things. I mean, I just don't know, how do you actually measure whether those things are in your, in your soil? How long do they stay? And I guess I'm just more interested in the idea of that biology being homegrown, really.
Yeah, no, those are really, really insightful comments. So one of the grant proposals that I have currently under review is teaming up with, a collaborator who looks at arthropod communities. So he's looking at like springtails and mites. And we want to kind of integrate arthropods and nematodes into the soil framework to be able to see like, is there kind of a keystone species or like a key indicator that we can point to and really link to soil health? And, to date, that's just been absent. So I really appreciate your comment of like, yes, we want to look at the entire soil food web. And that's one of our goals. And to your point about like, nematode communities, are they changing? How, at what ratios are the populations? So nematodes, the one thing I like about nematodes is unless your soil is like completely destroyed, you will see some type of nematode in that community. It could be that you just have a bunch of bacteriivores. Sometimes we get samples back and it's largely bacteriivores. And so then we can tell the farmer and say, hey, you have a really unbalanced soil food web. Maybe it could be good to up your organic matter inputs so that you are feeding the soil food web, leading to an overall more balanced nematode community. But yeah, I don't think it has to just be nematodes. I think there are other really important indicator species out there, and we just have to do the work. It's sadly been ignored for a long time within the soil health framework.
I'm probably gonna get beat up for asking this question, but regenerative ag is the new and upcoming thing. And there are now 3 people doing certifications, Liz Haney, Gabe Brown, I believe, and John Kemp as of late. Just curious on your thoughts on how they're going to be able to incorporate some of your kind of type of work and how we move forward with this kind of stuff. Because I agree with Rufus, it's the biology. We need to know the benchmark or the baseline of what a healthy soil looks like in different regions too, because all of our regions are so unique as well, right? So
Yeah, so I'm not super familiar with these different regenerative ag kind of certifications. But I would have to agree, like, the first step for me is really understanding how different parts of the soil food web are like actually like scientifically related to different soil health indicators. And then the next step is making sure that these different indicators are available and able to be offered through commercial testing labs, because that I think is when different certification programs can start to incorporate these metrics. Like we're still trying to get microbial respiration offered through commercial testing labs. And so there's just, I don't know what the kind of barrier there is for commercial testing labs. I did just see that like Spectrum Analytics is looking for a new technician that would start running permanganate oxidizable carbon and microbial respiration. So I'm hoping that that's a sign that some of these commercial labs are starting to run these things. But unfortunately, I feel like we have a long way to go with the soil biology. With that said, I want to give an example of work that's being done in the Netherlands. I actually went to the Netherlands to be trained in nematology, and my grad students go. For whatever reason, nematology is just way more advanced in the Netherlands and other parts of Europe. And they actually have diagnostic labs that have high throughput, you know, assays where they're able to get farmers nematode data very quickly. So I think that is the route that I'd love to see commercial labs in the United States go. And there is kind of a formula for that in other European countries. And so I hear your frustration though on wanting to know more about the biology. And I think like for whatever reason, we're just slower to get that moving here in the United States. There are
Well,
things.
Oh, sorry, did I cut
Oh
someone
no,
off?
I for the first time was able to look at the chat, but I don't see any additional questions.
Well, I feel like we could probably go on talking about this for quite a while. There's a lot to delve into, and I really appreciate, Christine, the way that you gave us this snapshot into just the incredible complexity of the soil. Thank you so much. Thank you everyone for participating and coming along today. And yeah, I look forward to more conversations about soil. Thank you.
Thank you, everyone. And again, reach out if you have questions, and also look forward to, you know, 3 months from now, hopefully we'll start being able to receive farmer samples. So if you're interested in soil health assessments, definitely reach out to us. Appreciate your time. Thank you.